This is Kelsey Springer's final exam speech for public speaking. Why I want to be a forensic scientist. So when I was in high school, I took a chemistry class that I absolutely hated, mainly because our teacher refused to actually teach chemistry. But for our lab portion, we learned the process behind forensic studies, such as hair, fiber, ink, handwriting analysis, and more. Our final was a game of Clue, where we applied these techniques to find the culprit. Mr. Mustard was found guilty, just as when I was a kid, but now with science. However, despite loving that lab, I believed chemistry was an evil subject that I could never hope to enjoy. Then I came to college, and everyone was so worried about having to take general chemistry, but I found that it was one of my best classes, and I enjoyed it more than my major subject, which was my Mar Bio class. I was marine science at the time, but I switched to biochemistry because I just enjoyed that chemistry so much. However, it wasn't until I was assigned a book called The Blooding in my genetics class that I refound my interest in forensics. And I'm going to tell you a little bit about The Blooding now. On November 21st in 1983, the body of 15-year-old Linda Mann was found just off of an old footpath in Narborough, England. She had been raped and strangled to death. Forensic scientists analyzed the scene of the crime, but the only piece of information they could find that linked them to the killer was the semen, but testing it at this time did not have the advantage of pinpointing it to one person in particular. Could rule out some, but still left a wide pool of suspects open. A group of officers went house to house, interviewing every male between the ages of 13 and 34, which was a number that was chosen based on sperm count found, and while another group of officers manually cataloged all men in the surrounding area with previous offenses. The hope was to link a male from an interview to a previous crime and help narrow the suspect pool. People were brought in for a blood test, but the best forensics labs could do with the information was say maybe they had done it, maybe they hadn't. This never-ending search continued for 3 years, and then the body of Dawn Ashworth was found in Enderbury, one town over from Linda Mann. She had been killed on July 31st, 1986, and like with Linda, Dawn was found to have been raped and later strangled. Again, door-to-door interviews were launched, and tips from the outside were investigated. A 17-year-old had been reported by a few people, placing him at the scene of the crime. He was arrested and confessed to the crime after a 48-hour interview. However, at this time, scientists were interested in studying the difference in people's genes, mapping them out, but no one had yet been able to mark individual genes. In 1984, Alex Jeffrey was studying the genes and how they evolved. While doing this, he was able to find a way to finally map them. By taking DNA found in blood cells and cutting them with enzymes before dropping them in an agarose gel with an electrofield, he could separate genes into smaller fragments. From there, he could use a southern blot to transfer them onto a nylon frame and use X-ray radioactivity to map each layer of genes. This produced a series of barcoded lines that showed the human genetic code. He went on to find that the genetic map was consistent despite the kind of tissue sample used, blood, semen, etc. He was able to see the variability in a person's genetic code and predict that it would be near impossible for two people to have the same genetic code, so he named this new process genetic fingerprinting. This new technique was a breakthrough in many areas, from birth defects to finding a person's parentage, but it especially became useful in forensics. A person can now be placed at the scene of crime with just a single drop of blood. So back to the murders. After the suspect was arrested and charged, Jeffries was asked to test a sample of semen from both crime scenes, as well as a blood sample from the suspect, in hopes that this would further solidify his case. Since he was a minor, it was pretty hard to charge him for both, seeing he was 17 at the time of Dawn Ashworth's, but he would have been 13 at the time of Linda Mann's, and that just didn't add up to a lot of people. So he analyzed the samples, found the genetic code of the samples from the crimes, as well as the sample from the suspect, and he had some good news and some bad news. He was able to prove that the two crime scenes did match up. They were in fact the same killer. However, the killer was not the suspect they had in custody. They did not match. This was the first time ever that genetic fingerprinting was used to free a murder suspect. An innocent went free that day because of science, a new breakthrough. After this, police launched a full-scale investigation based on genetic fingerprinting alone. Men from all over the surrounding area were sent a letter in the mail asking them to come in and submit blood sample and to test for genetic fingerprint matching the killers. They called this the blooding because over the course of the next year, blood samples were taken from 4,583 men in the surrounding areas of Norrborough and Enderbury. Still, this did not lead them to the killer until it was a tip that was called in by police about someone faking their identity during the blood test and having someone else submit a sample for them. Colin Pitchfork was taken into custody, wherein he confessed to both murders as well as 2 other sexual assaults. When his DNA was tested, it was an exact match to those found at both murder sites. He had previous charges for flashing. However, he had escaped suspicion before, after having a convincing alibi for the night of Linda Mann's murder. He escaped being prosecuted during the blooding because he faked an ID, and a coworker went in to submit a sample for him. He was given double life sentences for the murders, but it was given a minimum of 30 years. Alex Jeffrey's work went under a lot of scrutiny over the years, but it was here to stay. Other methods for mapping genetic fingerprints had been found, but it was his breakthrough that led to the eventual capture of Colin Pitchfork. Today, genetic fingerprinting is still used in forensics in court cases, as well as identification of a body. It is also used to discover a person's parentage, diagnose hereditary diseases, and more. In recent years, genetic fingerprinting has been used to differentiate between different populations of the same species and show how they varied from each other over time. This story reestablished my love for forensics that I had originally found in my high school chemistry class. And it was the power that it could have in a criminal prosecution that led me to think that maybe I could help. Maybe I could use my knowledge of science, use that love of chemistry that I now had found, and apply it to something in the law enforcement area. If it weren't for scientific breakthrough, a serial rapist would have never been found, and an innocent man would have gone to jail. In the years since, genetic fingerprinting has become a major way of finding a suspect with the National DNA Database established to keep information readily at hand. It's the effort of forensic scientists behind the scenes that fuel investigations such as this, wherein police work was not enough. Otherwise, they go cold, and scientists analyzing the scenes to find the suspect are all that can help it. Nerds can catch the bad guys too. I couldn't think of a better way to spend the rest of my life.